right, thank you very much. I'd like to thank the organizers for inviting me to give a talk here. I'm giving this talk on behalf of my collaborators, Ron and Anna Bromley, who are the founders and CEO and president of Biostat Solutions in Frederick, Maryland, and Ted Kalbfleisch, who's the founder and CEO of Intrepid Bioinformatics. And so what my talk addresses is basically there's a tendency for people who have, when I say people, I mean organizations, entities that have huge biobanks, huge genetic sample resources. There's a tendency to sort of stockpile these biosamples with the intention of possibly going back to them and doing something later. But the tendency is to sort of sit on the sidelines and wait until sort of the ultimate genetic tool comes along and everybody sort of points at next-gen sequencing and eventually being cheap enough that we'll just apply that to every sample and sort of be done with it. But what my message is that we really don't have to wait for the ultimate technology to get here to do that. We're sort of missing the boat by doing this for various reasons. So I'm going to get on to the talk. First of all, I want to give a disclaimer. I am clearly in industry myself. But the point of this is that I'm actually harmless. I'm the founder, I'm the CEO, I'm the only employee of Genomics GPS. And what my mission is, as I said a moment ago, my mission is to focus on the strategic application of these genomic resources to identify useful studies, to identify useful samples or sample collections. And then my own strength within this is the analysis of genetic data. I've got a long history in doing that. And so, you know, I'm working with, you know, anybody who sort of fits into that mold, and it doesn't necessarily have to be human, although certainly a large part of the data that I've looked at. Sorry? It helps. Well, you know, my collaborators typically are human, but the data does not necessarily have to come from humans. In fact, one of the projects I got now is non-human genetic data. Okay, so to give you a quick overview of what it is I'm going to try to do, I'm going to identify what the current process is, which basically is the sitting on the sidelines. I'm going to propose a change to that process, and then obviously compare the two. And what I'd like to get is, since this is a predominantly scientific audience, I want to get at the scientific benefits of this new model. And ultimately, to make it fly, it has to have commercial benefits as well. And then I should have plenty of time for discussion. Okay. So first of all, what do I mean by genetic biosamples? Obviously, this is an OMICS conference, so not everybody's focused on DNA. I am typically focused on germline DNA. And so typically these biosamples, these resources are blood often that's treated for clotting, stored away in a tube with antimicrobials, and typically frozen and sort of set aside. It can occasionally be extracted DNA. Again, it's frozen off, or could be blotted. So Whatman filter paper that's impregnated with my antimicrobials is a good mechanism of storing away DNA. And the desired data from these resources is typically a genotype or DNA sequence of one or more loci. It could be the subject of, for instance, a GWAS study or whole exome sequencing or sequencing or candidate gene sequencing, you name it. It's there as a resource. So the current model is to take the sample. So start with the patient or the subject, with the phlebotomist, or occasionally with the spit tubes. If you're certainly our colleague Professor Fernandraju mentioned 23andMe. That is a genetic sample. You can take this and prep it for storage, which means, you know, get ready to bank it away for months or years or eternity. But then, at some later date, once it's decided that this sample needs to be used, it needs to be retrieved. And then once it's retrieved, if it's been frozen, it needs to be prepped, again, quantified, checked, and made ready to do the genotyping. And then there's the actual process of doing the genotyping. Then whatever's left over is sent back to storage. So that sort of entails sort of the physical things that go on that require some combination of robotics and humans to do these assays. ultimately, the data are obtained, and then on top of this whole process is an inventory, sort of keep track of where this sample has been stored, how much of it is left, and what kind of format is it in. Is it frozen, or what kind of tube is it in, all that sort of logistical stuff. So the simple idea here is, rather than to emphasize all of this operational logistical stuff with the physical sample, is to take the sample and prep it immediately for genotyping. And, okay, what kind of genotyping will we do? And so I'm advocating that we do some sort of uniform genotyping, and I know for a fact that there are a number of distributors out there, and we got, in fact, have some companies in the audience that in fact will do genotyping, next-gen sequencing, what have you. But I know for a fact that you can, one of the current providers gave me a quote yesterday morning of $60 to $70 a sample for their biobank chip. So if you can do a biobank chip, which is one of the genotyping chips, that will give you on the order of 1 million SNPs, and then you follow that with imputation. So I'm not going to get heavy duty into what imputation is, but it's an algorithmic analysis that allows you to do indirect genotyping by basically taking the SNPs and the genotypes at flanking sites and allowing you to infer quite rigorously what the genotype is likely to be at any intervening SNPs that are in fact not genotyped. So this allows a combination of direct genotyping and indirect genotyping, that is the imputation, which will actually get you on the order of 10 million SNPs worth of data. That's a lot of data. Okay, so we'll come back to that. But the point is to do a fairly thorough job of direct and indirect genotyping at this stage, so we really don't have to worry so much about needing to retrieve this sample for subsequent genotyping. I do advocate that we, take a sample and a remainder of the sample and sock it away for storage. So if and when it becomes desirable, say, to do a whole genome sequence of that individual, or cheap enough to do that individual's whole genome sequence for whatever reason, we have an archival sample that will allow you to do that. And again, that need not be frozen. That might just be a sample on Whatman filter paper or something. So again, the data are obtained, and then, at this point, the inventory is simply, keeping track of this archive sample. So the pros and cons of what's going on now. Okay, currently, everybody sort of knows how to do the DNA biobanking. Stack it away. There's no upfront genotyping costs. So that, that's really one of the major impediments right now. It's like, why do the genotyping if we don't necessarily know what it is we want to get out of it? Okay, and so I will come back to that for sure. Okay, you obtain only the data that's desired. So, if you think there's only one gene you're going to be interested in, maybe, that's the focus of your study. But as you know, in science, you never necessarily know what all the questions are when you start off. One question always leads to more questions. And of course, playing the waiting game, genotyping and sequencing costs are continuing to drop. So, that's sort of a safe position to have. But the negatives in doing this are there's always the possibility the sample gets lost, or there's a sample mix-up. There's always human error. There's always the possibility of a freezer going down and you lose the sample forever. And certainly with humans, you have a finite lifespan of that sample. The consent can expire. Typically there's consent on the sample, but not necessarily on the data. So get the data while you can. Another negative is the expense. There's a considerable expense in all of the storing, retrieving, and prepping of the sample, getting it ready to do the genotyping and storing it away. And to me, the worst thing about this is the time lag. If you all of a sudden say, I've got 10,000 people socked away from a clinical trial, and the FDA says, hey, I'm interested in knowing what the genotype is at the following 3 genes, and you don't have that data, and now you've got to retrieve the samples, genotype the samples, and that can be a very time-consuming process. So the time lag involved in getting these samples genotyped can be a real issue. And then finally, it sort of galls me that you know, the people have been consented and given the sample with the intent that it be used for scientific research, and quite often nothing's done with the sample or very limited use is made of the sample when in fact it may have been broadly consented. And ultimately the data that are produced are very non-uniform. They're sort of, you know, very, very patchy. So what I am advocating is that if we in fact focus on doing the genotyping up front as the sample comes in, the data will be available immediately, more or less, and they'll be far more uniform. We'll completely get rid of the expense for storing, retrieving, and prepping the sample prior to the genotyping, so we can focus just on the genotyping. Again, we've greatly reduced the time lag in having access to the data. We've maximized the use of the sample, so there's no worries of the sample, you know, its consent expiring or what have you. We've avoided the possibility of actually losing the sample, you know, due to technical or other reasons. And also, with the idea of doing imputation, we've got this scaffolding of direct genotyping that has been done under this model. And what we know about imputation methods is they're continuing to improve. So there's actually quite a strong possibility that the data that you have from this sample will improve over time as the reference panels and the algorithms that are used for imputation continue to improve. You know, so you don't have to worry about not having gotten all the data, you know, right up front. Now the negative, the biggest one obviously, is that there's an upfront genotyping expense. If you use one biobank chip versus you know, the competitor's biobank chip, you know, you may miss the desired data. And then finally, you know, there is a much stronger requirement for an infrastructure for data maintenance and access than there is when you're just sort of doing a sort of a scattershot analysis. But I think if you're in this game, you really need to have that infrastructure for bioinformatics anyway. I think we all agree with that, given the nature of this conference. So what would perfect genomic data be? You know, certainly for germline data, we'd like to have the complete and accurate full sequence. We'd like it to, and we're getting close to that now. We'd like it to include structural variation, both the simple kind, like simple insertion deletion variation. We'd like it to include the more complex structural variation, although I'd have to say we're a long way from that with DNA sequencing methods. And as geneticists, we'd like to have the phase. The phase, you know, is accessible, and I think we are getting that with a lot of the imputation algorithms. So we're getting long haplotypes for many regions. What I'm not considering in this model is mosaicism, and I'm not considering epigenetic phenomena. So, you know, I'm sort of drawing the line as to what's sort of the best we can do. So what would be sacrificed in this proposal? Well, clearly de novo mutations. But if you think about it, when do you want to look at de novo mutations? That's typically more of a patient-focused exercise, and it's not the kind of thing that you're going to expect to see in large, you know, large biosample repositories. If you've got a single patient that has a relatively unique condition, sure, you're going to do the sequencing looking for de novo mutations that, you know, this phenotype doesn't match the parent. So that's sort of a different entity. Likewise, if you're looking at very private variation that runs in a single family, that's accessible through Mendelian methods, and you can, you know, pin down the region with the highest LOD score and with linkage studies and then sequence that. But again, that's not the kind of biorepository that tends to exist out there. And then any variants that fail to impute, whether they're rare or common variants or indels or CNVs, those will be less amenable to this program. But as I mentioned a minute ago, it's very clear that imputation algorithms are in fact improving. So there would be hope that these last categories will slowly disappear as imputation improves. And phase, as I say, is typically a part of the imputation process. So that will be accessible under this scenario. So what would be the commercial consideration of switching from the current paradigm of sitting on the sidelines and doing the genotyping right off the bat? Per sample, I think it's a wash. Certainly the upfront genotyping cost is one that people are not, at this point, have not faced, but I know that the reagents, and don't worry, I've only got a slide or two left. So, you know, what people are facing in terms of genotyping costs, if it's only $60 or $70 to genotype, just do it. Just do it, have access to the data, sock the sample away, because it's a wash. $60 or $70 is probably far less than the cost it would take to prep the sample, store the sample, retrieve the sample when you start factoring in, you know, the infrastructure and the personnel and the reagents needed for doing all of that. Okay. In terms of, you know, your emphasis now, it's on the data. It's not on the storage and retrieval of the sample. It's the storage and retrieval of the data. So this really sort of leads you to the next step, is the infrastructure. If you're not already in this and you haven't already made an investment, you know, in lots of freezers and personnel to run your biobank, starting into this, your needs for such infrastructure are now minimized. You know, so basically you might want to consider non-frozen archival samples, and the shift now would be to entertain a more robust system for data infrastructure. And so that's why my colleagues were on this with me. I like to think about this sort of thing, but my point is a lot of this stuff can be outsourced and is outsourced these days, whether you're doing the statistical analysis or the bioinformatics needs, or even the genotyping. A lot of this used to occur, you know, in just a handful of places because of the significant needs for all of this infrastructure. So that's it. I'm open for discussion. I don't know if that shows up, but that's my trio of rescue dogs, the ends. All right, Krishna.
Sure, go ahead.
What I want not clear to me is, have you answered your own question at the beginning? How do you sample your various sources? What is the method of sampling? Or, I mean, I should say methods of more than one method maybe perhaps.
Right, right. So obviously it depends on what the question is. Like, what is your ultimate question in coming up with this biobank in the first place? So I've thought, I guess most of my audience that I've thought of in terms of this would be like pharmaceutical companies who are doing their clinical trials, and their sampling is pretty well laid out in terms of who they're going to recruit into their clinical trials. You know, obviously if it's phase 3, they're recruiting people who have a disease and are taking the medication. You know.
That begs another question, which is, what is your goal? So the sampling has to meet your, yeah.
Right, right, right. And in terms of, you know, sample size, and typically, you know, for people who already have biobanks, obviously I've not been privy to what those questions were and why they felt the need to have a biobank in the first place. But there are entities that are, governments for instance, that are interested into doing genome projects for their entire country. And, you know, they are in fact sampling. So I think it is important to make sure. I know India had a big Indian genome project where they did sampling. I think Partha was guiding them on that. I don't know, perhaps you were involved as well.
Yes. And a country like Iceland, for example, Iceland has its own project which is pretty comprehensive, I believe.
Well, Decode, of course, has all the electronic health. Well, they weren't electronic initially. I think they've all been converted to electronic.
I have one more question.
Sure.
This, you are excluding mosaicism. Mosaicism is excluded, I noticed at the beginning.
Yes.
Aren't you eliminating some really valuable information by doing that?
Well, my thinking, number one, I was looking for germline DNA. And, you know, what can we hope to get in terms of, if mosaicism is there, we would certainly want to know that, but it's kind of in the nature of the blood sample. So for epigenetics can be tissue specific, and if everybody's got a blood sample, you know, you've got 100,000 blood samples, epigenetics may or may not be important. For mosaicism, I was thinking very similarly, you know, if if we're looking, for instance, at somatic variation in cancer, we are typically more interested in doing the sequencing. If we're interested in low levels of mosaicism in blood, that could be important, but it certainly was not, wasn't my main line of thought. Well, all right, what would you hope to get out of a pre-existing, 10,000 subject biobank in terms of gonadal mosaicism? I mean, if their blood samples socked away and they have a certain level of phenotypic information, or say there's a project coming up to collect patients for a big GWAS study in heart disease. To what extent can you, collect blood samples and hope to have anything important to say about gonadal mosaicism? Would you? Okay. Yes, Jeff.
Hi, yeah, good talk, thank you. I was just curious in terms of the scope of what you're proposing. Were you thinking of all stages of clinical trials or mostly phase 3? I guess I'm thinking, phase 1 where you have healthy volunteers, and we do collect samples, but generally unless there's outliers for PK or for exposure or safety, we don't tend to test them.
Yeah, I mean, my thinking is if the genotyping costs are getting low enough, and if you're going to be spending on the order of 60 or 70 bucks, even for a healthy volunteer's phase 1 sample, just genotype it, and hope that you, and then archive a small sample. And if in fact the person's an outlier, you check the genotypes and say, okay, is there anything, if I suspect HLA, if I suspect one of the ADME genes in this person being an outlier, you check that first, and then if you say, well, I'd really like more resolution for the ADME genes, say, then you could in fact genotype that subject. But you have a scaffolding of genetic data with the hopefully extensive imputation to start with. So and you'd have the data immediately, so having it right up front, because you know that if you've socked the sample away and you've got this one outlier, then you have to go to the biobank and say, please retrieve this sample and prep it for genotype and genotype. I mean, the question may be urgent and you're not necessarily going to get that data urgently. So it's good to have the data up front, even for phase 1. Okay, I think that was the end of my time.
Yes, sir.
So on that, let's see, I guess I'm supposed to do this. Wait.